It is well established that ionizing radiation is a health hazard. In this video, I will discuss the biological consequences of radiation, in particular DNA damage, and its implications for diagnostic imaging. Photons from the X-ray beam interact with the patient's tissues. In particular, Compton scatter and photoelectric absorption both cause ionization. The electrons that are ejected during this ionization process cause subsequent ionizations and are the mediators of energy deposition. Although these ionization events can occur in any biological macromolecule, ionizations that occur in the DNA are particularly relevant. DNA damage can result in cell death, cancer, and heritable mutations. Thus, DNA damage is considered the target for radiation-induced biological outcomes. Now let's take a closer look at radiation damage caused by DNA. Approximately one-third of the DNA damage occurs because of direct actions, where the ejected electrons directly ionize the DNA to form unstable free radicals. These free radicals quickly reform into stable configurations by dissociation or cross-linking. In contrast, in indirect action, the ejected electrons interact with water to form unstable water radicals, which subsequently ionize DNA. The chemical reaction of X-ray photons with water, referred to as radiolysis of water, yields highly reactive radicals, including the hydroxyl radical and the hydroperoxyl radical. Additionally, hydrogen peroxide, a strong oxidizing agent, is also formed. Because the radicals are highly reactive, they will diffuse limited distances within the cell. It is estimated that these radicals need to be formed within a diameter of 4 nanometers around the DNA in order to be able to ionize DNA. So, to summarize X-ray photon action, initial action of X-ray photons via Compton and photoelectric interactions result in ejection of electrons with kinetic energy. These electrons will deposit energy within the cell. When this energy is deposited within the DNA directly, it is referred to as direct action. In contrast, when the secondary electrons interact with water to produce free radicals that subsequently damage DNA, that mode of action is referred to as indirect action. More than two-thirds of the DNA damage from X-ray photons occurs via indirect action. DNA damage is the primary cause for radiation-induced cell death, heritable mutations, and carcinogenesis. Ionizing radiation produces different types of DNA damages, including base damage, single strand breaks, double strand breaks, and DNA and protein crosslink. Mammalian cells have evolved elaborate mechanisms to respond to DNA damage. This includes sensor molecules that recognize specific DNA damage types and activate signal transduction pathways to upregulate DNA repair mechanisms. Most base damages, single-strand breaks, and DNA crosslinks are efficiently repaired by base excision repair and nucleotide excision repair mechanisms. In contrast, double-strand breaks are less efficiently repaired and are thought to be the detrimental event for cell killing, tumor induction, and heritable effects of ionizing radiation. DNA double-strand breaks are repaired either by homologous recombination or non-homologous end joining. Non-homologous recombination is an error-prone process and is thought to be responsible for many of ionizing radiation-induced mutations. In eukaryotes, the DNA is distributed into chromosomes. Each chromosome is a long DNA molecule associated with proteins that fold and pack the DNA into a compact structure. During the S phase of the cell cycle, the chromosomes are duplicated and the two daughter sister chromatids are held together at a region called the centromere. During mitosis, the sister chromatids separate at the centromere and are segregated into daughter cells. Now let's consider DNA strand breaks that occur prior to chromosomal duplication, that is in the G1 or in the early S phase of the cell cycle. In this case, the damage is replicated and both sister chromatids will carry the damage. This aberration is called a chromosome aberration. Next, let's consider irradiation after chromosomal duplication. In this case, the chromosomal break occurs only in one of the sister chromatids, and this is termed a chromatid aberration. Some aberrations are lethal to the cell. These include the ring chromosome, the dicentric chromosome, and the anaphase bridge. These aberrations impact chromosome segregation and cause cell death during the next mitosis. In contrast, non-lethal aberrations do not cause cell death. However, as a consequence of the chromosomal aberration occurring in a somatic cell, could result in neoplasia. If these aberrations occur in the germ cells, they could cause heritable disease that is manifested in subsequent generations. So, to summarize the consequences of DNA damage, radiation induced DNA damage may be non lethal, resulting in DNA mutations, or alternatively, it can cause lethal damage that results in cell death. Based on the outcomes of the DNA damage, radiation effects can be categorized into two fundamental categories: stochastic and deterministic. Understanding the difference between these two categories of effects is of particular importance to radiation safety and protection. Stochastic effects are a consequence of sublethal DNA damage. Importantly, stochastic effects are considered to occur without a dose threshold. This belief is shaped by our current understanding of the molecular mechanisms of DNA damage and repair. A single X-ray photon has the potential to cause a DNA mutation. Thus, even the smallest radiation dose could cause a heritable effect or a cancer. As the radiation dose increases, the number of radiation induced DNA damage sites increases, and the risk for subsequent disease causing mutations is higher. Thus, the probability of stochastic effect increases with dose. Radiation induced cancer and heritable effects are both stochastic effects. In contrast, deterministic effects of radiation are caused by cell killing and are the consequences of cell death on tissue or organ function. Deterministic effects manifest only when the radiation exposure to an organ or tissue exceeds a threshold level. The magnitude of this threshold is dependent on the type of tissue. When the dose is below the threshold, the effect does not occur. Exceeding the threshold dose will cause an effect in most of the individuals exposed. As the dose increases, the severity of the effect increases. To understand the difference between deterministic and stochastic effects, let's consider the effects of ultraviolet radiation in sunlight on the skin. One effect is skin burn, which is a deterministic effect. By keeping our sun exposure below a certain threshold, we can completely eliminate the risk of getting a sunburn. Another effect of ultraviolet radiation on the skin is radiation induced cancer. This is a stochastic effect. By minimizing our sunlight exposure, we minimize our risk of radiation induced cancer. However, given that we will all get some amount of sunlight exposure, we cannot completely eliminate that risk. So, the distinction between stochastic and deterministic effects is particularly relevant for radiation safety. By keeping our doses below the threshold, we can completely eliminate the risk of any deterministic effects. And by keeping our doses as low as possible, we can minimize the likelihood of stochastic effects. Next, let's consider the temporal sequence of these events. Note that the ionization and the initial molecular damage occurs within seconds to minutes. Importantly, note that the outcome of this damage can manifest in a few days or in many years. As described, the principle of diagnostic imaging is to keep the doses below the threshold for causing deterministic effects. Thus, deterministic effects resulting from cell death are not a concern with diagnostic imaging. Additionally, several studies have demonstrated that heritable effects, especially with diagnostic dentomaxillofacial imaging, is negligible. Thus, the only risk from diagnostic dentomaxillofacial radiology is the risk of radiation induced cancer. And given that radiation induced cancer is a stochastic effect, it impacts how we apply diagnostic imaging in practice.